The immune system is divided into 3 different lines of defense. So there are 3 different lines of defense in the immune system. Again, everything in the immune system is really trying to prevent stuff from getting in the body, and if something does get in the body, it's let's get rid of it from the body because it's something foreign. And so the very first line of defense are all the ways of how can we just prevent certain microorganisms from getting in the body. Obviously, we don't want these bacteria in our body. We don't want viruses in our body. We don't want fungus or parasites or anything else in our body. And so it's looking at just different physical barriers of preventing things from getting in, such as the skin and the mucous membranes. The second line of defense are all the different things that our body can do to these organisms once they do get in the body. But these are all, they're called innate. Innate just means things we're born with. So these are not learned things. We have cells in our blood, in our body, that know how to kill. They weren't trained, they just knew how to kill. We have phagocytes that their job is just to eat. They were not trained to eat. They just know how to do that. There are just certain cells that know how to do certain things, regardless of what got in the body. There's different chemicals in our body that can help with getting rid of things. There's different processes like fever and inflammation. These are different ways of trying to get rid of that pathogen. It's not for any particular specific pathogen. It's just things our body was born knowing how to do. And so the first 2 lines of defense of preventing things from getting in the body and then things our body just knows how to do and trying to get rid of some of these foreign invaders are both called the innate immunity. All things our body was just born knowing how to do. Our body was born with skin and mucous membranes. And because that's chapter 14, because those first 2 lines of defense are a little bit easier to explain. I mean, the skin is a little bit of review from A and P, and it's, I would say it's not a tough, complex idea of how it prevents things from getting in. Even some of the different cells and processes like inflammation and fever and phagocytes, not super complex in the whole process on how they work. That is all part of the online portion because this class is still partially online for lecture. And so chapter 14, your job is to watch that video on your own and take a quiz. So if you look at the calendar, which I didn't bring up, the calendar is chapter 14, the innate immune system, is watch that video. I think it's an hour. You literally get through all of those 2 lines of defense in an hour and take that quiz by tomorrow night, midnight. So we'll just put that on the calendar. Don't forget to go through that first 2 lines of defense. The third line of defense, which is chapter 15 in the ebook, is definitely more complex, and it's our adaptive immunity. This is where we can recognize very specific invaders that get in the body. We can form memory cells, and we specifically talk about a type of cell called lymphocytes. It's a type of white blood cell. Way more complex, which is why I didn't want to put any of it online. So the plan is that we will get through all of chapter 15 in lecture face-to-face, hopefully. So that's kind of how I divided up. Chapter 14, a little easier, put it online, you can make it through it, not too complex. Chapter 15, I'm going to draw stuff out and try to explain it, because it is probably the most, I hate to say it, because I'm like, it's confusing. Then everyone's like, oh my God, I already hate it. But there's a lot of steps that go on. It is truly like full processes on how our body recognizes things and how it can get rid of it, and then how it can form memory cells. It will take us definitely not today. We will finish this PowerPoint. I'm hoping within Thursday or Wednesday we will get through it. So, it was working. Another demonstration of how the immune system is broken up, those 3 lines of defense. The innate defenses, those are all things that our body was just born knowing how to do. So the first line of defense, the surface barriers, the skin and mucous membranes. The second line of defense, Again, those are examples of different things that our body is just born knowing how to do, and that's all of chapter 14, fever, inflammation, phagocytes, different types of chemicals like proteins. So this PowerPoint, we're going to go over what is the adaptive immune system, where does it all take place in our body, what are the main players, like what are B cells, what are T cells, what are antibodies, what are cytokines, what are something called MHC and APC, and we're going to go through the cell-mediated immunity. It's one process on how to get rid of foreign invaders. The humoral immunity, which is a whole other process on how to get rid of invaders, how they do chat with each other, the different processes, and how we can acquire immunity to different things. We won't get through all of it today, though. So, what is adaptive immunity? So, it's the body's ability to recognize and defend itself against, say, foreign invaders. That's the other word I wanted in there. Foreign invaders and their products. Immunity. There are 5 attributes. The first one is the specificity, the fact that our immune system can recognize very specific invaders. Like there are millions of different things that our body can recognize, and it recognizes each one as its own individual thing. So our body can recognize very specific foreign invaders. We can recognize every virus, every bacteria, every fungus. We can recognize different types of venom. We can recognize just, anything that could possibly get in the body. Our body can recognize it specifically. It knows how to do something about it. Inducibility. I'm going to write some stuff up here. Recognizes that very specific foreign invader. Inducibility means, there's a short way to write it. The foreign invader, or pathogen, I guess it's not always a pathogen, induces cells the adaptive immune system. When we say induces, I'm really looking at activates. That's what it means. The cells in our adaptive immune system are just hanging out, just waiting to recognize these millions of different foreign things that might get in our body at some point in our life. But it's once they recognize them, now they become active. So this foreign invader, once it gets in the body and it's recognized, it now activates or induces these cells of the immune system. And then the clonality is once induced, once activated, the cells generate clones of themselves. Have a couple cells in our body that are just circulating, waiting for some random foreign thing to get in our body, some random bacteria, some random parasite, whatever. Once it recognizes it, it becomes active. It's kind of like, hey, that's the thing I'm supposed to recognize. I need to do something about it. And the best way to fight off something is if you have a huge army behind you. And so it starts to make a bunch of clones of itself. One cell fighting another cell might not win the battle. But if you can make clones of yourself, and now you've got 1,000 of you, fighting that one particular cell, the more likely to win the battle. So it makes clones of itself, so it forms this nice, strong army to fight off the foreign invader. Unresponsiveness to self, kind of explanatory, is that our adaptive immune system has to be able to recognize normal body cells so that it does not respond to our normal body cells. We want our adaptive immune system to recognize foreign invaders or some type of abnormal cell in our body, but we do not want it to recognize our own normal body cells, or it's going to mount an immune response against our own normal body cells, which is not good because then it's going to fight off our own cells that are perfectly happy and doing wonderful things. And this is when we get into different types of autoimmune diseases, is when our immune system starts to fight off our own body. It's not normal, and it's not good, and so our adaptive immune system is supposed to not recognize our normal body cells. And then 5, one of the coolest things about the adaptive immune system is that it can form memory cells. So once it knows how to fight off a foreign invader, the next time that foreign invader gets in the body, it remembers how to do it, and so it's a whole lot faster. The first time our immune system recognizes something, becomes induced, makes clones of itself, and then goes and fights the battle, can take sometimes a couple weeks, which is the time period you're sick. But the second time you get exposed to something, it remembers how to do something about it, it makes clones of itself a whole lot faster, and it fights off that foreign invader a whole lot faster, and instead of a couple weeks of it taking to fight that war, it can be literally within hours, which means it fights it off before you didn't even know you had that thing in your body. You wouldn't show any symptoms of it whatsoever. Now, the main players are lymphocytes, which are a type of white blood cell. The other white blood cells, other than lymphocytes, are all part of our innate immunity. So it goes over that in chapter 14, the eosinophils, the monocytes, all of those. But lymphocytes, one of our 5 white blood cells, is the main player that's going to be doing all of this in the adaptive immune system. But there are 2 types of lymphocytes. Under a microscope, you'd never be able to tell the difference. They look identical to each other, but they are structurally different from each other. And the 2 main lymphocytes are B lymphocytes or T lymphocytes. But lymphocytes is just a long word to say, so we usually just call them B cells and T cells. And where the B and the T come from is where the cells mature in the body. So the B cells, or B lymphocytes, they mature in the bone marrow, which is where they get the B cells. And the T lymphocytes, or T cells, they mature in the thymus, which is where it gets the T. Again, underneath the microscope, you'd never be able to tell the difference. But they do different things. And they are different from each other. So the adaptive immune system, so chapter 15, there are 2 different ways that our adaptive immune system can fight off foreign invaders. There's 2 branches. There's a humoral immunity method of getting rid of these foreign invaders and still getting memory cells. And there's a cell-mediated method of getting rid of foreign invaders and still getting memory cells. And they use different lymphocytes. One uses B cells, one uses T cells. So our adaptive immunity has 2 branches, and my hope is to get through our first branch today. So the first branch is called the humoral immunity branch. So this is going to stay up here today. And the other, which my plan is to get to on Wednesday, is the cell-mediated branch. And they use different cells. The humoral immunity branch uses specifically B cells. The cell-mediated branch, or cell-mediated immunity, is going to use T cells. Now, we'll get to it hopefully today. But the B cells are the ones that are going to be making those antibodies. A mediated branch, again, uses T cells, and there's more than one kind of T cell. I'm not going to go into it today because that'll hopefully be Wednesday's lecture. But there's more than one type of T cell. There's more than one type of T lymphocyte. B cells, there's only one kind of B cell. We call them B cells. T cells, there's different types depending on what they do, what their function is. So, the terms in adaptive immunity are antigens. So, antigens are molecules that the body recognizes as foreign. So an antigen itself is just anything the body recognizes as foreign. That's the official definition. But it's what about that antigen does the body recognize as foreign? Those are what are known as epitopes. So epitopes are little 3D structures, which I can't draw 3D very well, on the surface of the antigen that the body recognizes as foreign. I'm going to refer to it in the coming slides. So the whole thing is something that the body's going to recognize as foreign, but it's the fact that this big whole thing, this big whole antigen has little reddish color, I've read, little red triangles and little orange circles and little green squares. Those are the little 3D structures that the body will actually recognize as foreign. And so we have different cells that can recognize these little triangles. We have different cells that can recognize that little orange circle as foreign. We have different cells that can recognize that little green square as foreign. So our body could recognize every single little individual epitope on the surface of every possible antigen. And it's those 3D structures that are almost like the red flag on the outside of that structure that are like, I'm foreign. Which white blood cell is going to recognize me as a foreign molecule. This is just showing another book picture that the whole thing is an antigen, this whole structure, but it's these different little epitopes are the little 3D structures that our white blood cells are actually going to be able to recognize, attach to, and are usually the things that are going to activate those white blood cells. Now, different things that are considered antigens, I've got a list, I'll go look it up, but again, it's anything that's in the body that's foreign, that we normally wouldn't have. Things that are always going to trigger our body as a foreign object, peptidoglycan, that structure that's in a cell wall. We do not have anything that looks like peptidoglycan in our cells, so it's a guarantee that will always be a red flag to our immune system. But it doesn't have to, I was going to say, it's pretty much anything on the surface of bacteria, even flagella. I know that sperm has a flagella, but it's structured completely different than a bacterial flagella. So flagella is something that would be a big red flag that it's a foreign object. Anything on the surface of a virus is going to be a big red flag. It's going to have very distinct, unique epitopes that our body's going to recognize as foreign. But it doesn't have to be bacterial. It doesn't have to be viral. It doesn't even have to be a parasite or a fungus as something that our body is going to recognize as foreign. Dust, all by itself, our body could recognize as foreign. Different foods have structures on them that our body could recognize as foreign. This is how we have allergies. If you're allergic to a certain type of food, it's because there's something on that food that our body recognizes as foreign, and our immune system is like, oh, we don't like it, we have to get rid of it out of the body and form a whole immune system response against it. People are allergic to dust, people are allergic to certain types of drugs. Again, those things all have some structure on the outside of it that our body recognizes as foreign. Do you guys have allergies to something? I want to say certain drugs. I'm allergic to shellfish. What? A lot of people are almost allergic to like gluten, that have the gluten allergies. Gluten is a pretty big protein, and a lot of times, generally, the bigger the structure, the more likely it is that it will get recognized as foreign. So it's because it's a really big protein. People are allergic to a lot of different proteins. That's why even proteins in peanut butter, why people have peanut butter allergies. It's a pretty big protein, and that pretty big protein is a big, huge epitope, and the body's like, foreign, now you have allergies. That's because your body recognizes as foreign, where other bodies are like, oh, that's food. That's what we like. We're showing that antigen, so again, it's anything the body recognizes as foreign is the official definition, but we do group, because there's so many different types of antigens, we group them into 3 types: exogenous antigens, endogenous antigens, and autoantigens. So the word antigen comes from antibody generator, smush it, because these are all things that are going to trigger our immune system. But an exogenous antigen, again, if we look at the prefix exo at the beginning of something, exo means outside, like exit, outside. An exogenous antigen, these are structures on the outside of an object that our body recognizes as foreign. So there are little tiny structures on the outside of these foreign objects that our body's like, yep, we know what that is. So if this is a bacteria and it's got a flagella on it, there are structures on the outside of that bacteria that our body recognizes. There's structures on the flagella that our body recognizes. What could be some other type of, extracellular microbe on the outside of cells that our body recognizes little outside structures. Very easily recognized. Endogenous means these are organisms that live on the inside of our cells, but are presented on the outside. We have a lot of bacteria that live inside of our cells. Viruses have to get inside of our cells to hijack them. And so if they're inside of our cells, our body has to still look at our cell and be like, there's something foreign inside of it. And our cells are not see-through to our adaptive immune system. And so our body cannot look inside of a cell like we can on this picture and be like, look, that cell has a virus in it. Instead, that virus that has little blue circles all over it presents itself on the outside. It was almost a little red flag. Those little blue circles from the virus are actually going to be on the outside of an infected cell. And so it's called an endogenous antigen because it's a virus or some structure that's inside the cell that's presented on the outside. So that our immune system now sees like, oh, there's little blue circles on the outside of that cell. That is not normal. That's a viral structure, and it will target that cell and it will kill it. And then the last group are called autoantigens. Auto means self. So if you have an automatic car, it shifts by itself. An autoantigen, these are self-antigens. So these are structures on the outside of our own cell that the body needs to know are normal. So although they're called antigens, which means they're recognized as foreign, and we call them autoantigens, as these are structures on the outside of the cells that we need to know are not foreign. Almost like an oxymoron to say an autoantigen, if it's an antigen, but it's on our own cells. So our cells have structures on the outside of it. We got carbohydrates on the outside of our cells. We got proteins that are sticking out on the cell membrane. And our body has to be able to recognize every single one of those structures and know that that's normal, not fight an immune response against it. So on the outside of our cells, these little structures that our body has to be able to recognize as foreign, some of them are just sitting on the outside of our cells. Some of them are presented on the outside of our cells. And we call them major histocompatibility complexes. MHC, shorten it because that's a big huge mouthful. So these are antigens found in the membranes, in cell membranes, in the phospholipid bilayer. So these are antigens found in the cell membrane of most cells of vertebrate animals, and they are not recognized as foreign. So these are things, these little blue things are different shades of blue, or these different things, different columns, different books, different pictures. These are things that are in our cell membranes that our body doesn't recognize as foreign. But they do have a job. They're not there just to make our cells look pretty. They do have a job. And it says they're found on the membranes of most cells of vertebrate animals. We are going to find these different types of antigens on every cell in our body except red blood cells. And the main reason why we don't find these on red blood cells is red blood cells don't have a nucleus. So really they're found on all nucleated cells in the body. And red blood cells are the only cell in your body that don't have a nucleus. They did at one point, but they don't anymore. Now, some cells in our body have both major histability complexes, and some just have one. So some are just going to have class 1, and some are going to have class 2. Class 1 are found on every single cell in your body, except the red blood cell. So every cell in your body right now has a class 1 antigen on it, except the red blood cells. Class 2 are found on antigen-presenting cells. So if you printed this PowerPoint, it's nice because you can draw an arrow like MHC class 2 are found on antigen-presenting cells. So every cell is going to have a class 1. So antigen-presenting cells have a second one. They have got both. They've got the class 2 as well. Examples of antigen-presenting cells, dendrites. These are not part of the nervous system. These are found in a lot of our skin. They just have lots of extensions on them. Macrophages, the big eating cells, B cells have them. But the idea for an antigen-presenting cell, whether it's a dendritic cell, a macrophage, or different B cells, these different cells eat objects, and then they present them to our adaptive immune system. To be like, hey, I just ate this foreign invader. Here you go. Here's some antigens. Do something about it. And they present these cells using that class 2. That's why they're called antigen-presenting cells. And they're like, oh, you didn't see it? Here, let me present it to you to do something about it. So every cell has a class 1. And class 2 also have a bonus one. Those antigen-presenting cells have 2 of them. Only because you're going to see these come up in some of my slides coming up, so I wanted to find them. Now, the humoral immunity, which is what we're going to focus on today, all has to do with B cells. So they arise and mature in the red bone marrow. Again, it's where they get their name. We find the majority of our B cells. We find them mostly in the spleen, the lymph nodes, and something known as your mucosa-associated lymphoid tissue. So this is lymphatic tissue, so it's part of our immune system tissue that's found near mucosa membranes. Now, just an example of where you would find a lot of this is our digestive tract. Our digestive tract is lined with mucous membrane. Our digestive tract, your intestinal tract, or your intestines right now, are jam-packed full of bacteria that are helping break down food, which is fine. But because we have bacteria in our intestinal tract, bacteria in our body. And we need to make sure we have a lot of this lymphoid tissue, this immune system tissue, kind of monitoring to make sure those bacteria don't cross the intestinal tract and get elsewhere in the body. So we have this lymphoid tissue right there where we know we have lots of bacteria. We call it mucosa, the mucous membrane, associated lymphoid tissue. And so it's another place we're going to find a lot of these B cells. We have a very few number of B cells in our blood. Most of them are hanging out in these various types of lymphatic system organs, the spleen, lymph nodes, and this lymphatic tissue. And the major function of B cells is the secretion of antibodies. Now, COVID did horrible things for us, but the one good thing COVID did for us is it's amazing how many people know more about the immune system and even immune system terms like antibodies since COVID happened. people are like, I don't know, right away it was like, oh, once you have it, you have antibodies, and you're harboring antibodies, and antibody therapy. Most people before probably never said the word antibody aloud. And then all of a sudden it became part of conversations. And so, that's good news, for learning about your immune system. And the B cell's main job eventually is to make antibodies. So again, the humoral immune branch, which is what I want to get through today, we have B cells, your B lymphocytes, and their main job is to make these different types of antibodies to fight off our foreign invaders. Now, a B cell. I'm not artistic, so just take everything I draw with a grain of salt. You know, here's a B cell. As exciting as I get, but I'm going to draw 3 of them. Now, every B cell has a B cell receptor on the outside. It's a receptor that's looking for a very specific epitope. Now, each B cell only makes one type of B cell receptor, so I'm going to use it color-wise. So this B cell is only going to make, and B cell receptors look like Ys. This particular B cell is only going to make red Ys. And this B cell is only going to make green Ys. And this B cell is only going to make brown Ys. So each B cell makes one type of B cell receptor. I color-coded it. And each B cell makes many copies of the same B cell receptor. You know, whether you have 5 on there or whatever. But it's got lots of receptor on it. And each B cell receptor could recognize just one thing. So this little brown one can only recognize one thing. This green one can only recognize one thing. This red thing can only recognize one thing. They're very specific. I chose these colors because this red B cell receptor, its job is to recognize red triangles. Whereas the brown B cell receptor, its job is to recognize, well, you know, my picture was orange, but brown circles. And this B cell receptor is very specific and can only recognize little green squares. So they're very specific in what they can recognize. One thing only. But they do have lots of copies of themselves. It has the ability to recognize millions of different epitopes. So I drew 3 different B cell receptors or B cells with 3 different B cell receptors to recognize 3 different little antigens or epitopes. We have millions of different B cell receptors in our body. We have millions of different B cells. And each of those millions of different B cells each have their one single B cell receptor that's just waiting to recognize a very specific foreign invader. So I always like to think B cells are kind of like snowflakes. You know, they're all unique. We've got millions of different B cells, millions of different B cell snowflakes in our body. Each one is unique. Each one is waiting for its own epitope to get into the body for it to recognize. Now, I zoomed in picture. So here's the cell membrane of a B cell. And it has this structure in it. This is the B cell receptor. Again, it does look like a Y. And it's at the very top of that Y, at the very top of the B cell receptor, that it recognizes something very specific. So at the top of this Y, it can recognize just little triangles. It's shaped. I always like to think of my B cell receptor as the big Y, and it's the hands, kind of like those big floppy guys. It's the hands that actually can recognize very specific things. So these are the hands, and the hand can only recognize one very specific epitope, little triangles. It's shaped specifically for, but it can only recognize the little triangles. This would be a shape that it could only fit little squares right in there. This would be shapes that it can only fit little circles. So that's how it recognizes them. The very hand part of the B cell receptor is kind of the action area, the very specific area that's looking for those very specific epitopes and can fit it and touch it and be like, I see you, I can bind to you. We'll become induced then, which is where we get into our next slides. The B cell becomes active because right away they're just those million, I'm just going to say million, those million different B cells are just floating around in your body. Again, they're hanging out mostly in your spleen. But they're hanging out in the body. They only become activated when that antigen, that very specific epitope on the antigen, binds to the hand. Now it's an activated B cell. Once I'm going to say we just activated this red one. And these tend to become activated too if this was in the body. But we've activated this B cell, and the B cell's like, oh my goodness, there are red triangles in the body, and I need to get rid of them right away. It will start to make more of itself. It's going to start to make more of this particular B cell. And all of a sudden, you're not going to have just one B cell in the body that has red Ys all over it, or B cell receptors. You're going to have thousands of the same B cell with the same B cell receptor, and they can all recognize little red triangles. So on this picture, this is showing 3 different B cells. They all have a unique B cell receptor. This one's got a green B cell receptor, it has like a dark blue, and then has a light blue B cell receptor. Only one of them recognizes this antigen. And in this case, it's the one that has the dark blue B cell receptors. Once it recognizes and binds to that antigen, it becomes activated and it starts to make more clones of itself. Now, instead of 1, we have 2. And yes, we're going to have thousands, but in the picture, it makes more of itself with the exact same B cell receptor. It's going to continue to grow and divide, and it's going to become different kinds of cells called effector cells, which is unique to B cells. They're going to change from this structure to something else. And they're called effectors because those effector cells are going to do something. More words. One of the effector cells that it become are plasma cells. So these B cells are going to grow and divide, and one thing they can become are plasma cells. Most of them will become plasma cells. In the picture, they're like, most, 3 out of 4. I'd say even higher than that percentage-wise. But most of them are going to become plasma cells. And it's the job of these plasma cells is to churn out antibodies. So by itself can't make antibodies. It first has to become activated. It has to start making more of itself. And then it has to change into a plasma cell to then start churning out these antibodies. And it's those antibodies that go out into the bloodstream and can recognize that same thing. Now, I think I've got a bigger picture of this. Here we go. So again, we only activated the dark blue B cell receptors with that antigen. It made more of itself. And then it started to make those effector cells. One of them, the majority of them becoming plasma cells to churn out antibodies. What do you notice about the shape of an antibody to a B cell receptor? They look identical to each other. An antibody looks identical to a B cell receptor. And they do. The idea is a B cell receptor is formed to recognize a very specific thing, little red triangles. Well, this antibody that is now made needs to be able to recognize that very thing that activated them. So all the antibodies that this thing, if it was a plasma cell, could make are all going to look like little red triangles. Again, they are little red Ys because it has to recognize little red triangles. These antibodies are shaped exactly like the B cell receptor, and they're shaped that way so that it can recognize that same exact antigen. So antibodies look identical to the B cell receptor because they have to recognize and attach to the very same thing. But not all of them become plasma cells to churn out antibodies. a few of them do become memory cells. And these memory B cells hang out in our bloodstream, they hang out in our immune system, and they can recognize these antigens, become induced, can make plasma cells, and churn out antibodies at a much faster rate. Again, it can take a couple weeks to attach, do that clonal selection, make more of itself, become effector cells, and then churn out antibodies, and those antibodies to go out and kill and get rid of whatever foreign invader it is. This whole process can take a couple weeks. But a memory B cell recognizes it, can do this, sometimes within hours. Yay for memory cells. Now, a little bit more about the antibodies. The antibodies still look like a big old Y. And they still have to have at the very, very top of the Y the little hands, and those hands are formed exactly like the B cell receptor so that it can still bind to that same antigen. This one is shaped to bind to little circles. That one is shaped, I don't know, like half triangle, half square type of shape. But they both have an antigen, they all have antigen binding sites to recognize very specific things. Antibodies are proteins. And so as some of you I know are going into nursing, when you explain to patients, like, why do you need to have a lot of proteins in your diet when you are sick? Antibodies are proteins. If you are lacking protein in your diet, that does affect your immune system. You can't churn out those antibodies quite as fast or as effective. So they are made up of antibodies, but antibodies have other names, so sometimes I use them interchangeably, and you'll see it interchangeably. They're also called immunoglobulins. And so I know on some of my slides, because the book refers to both of them as well, you'll see it just shortened to IG for immunoglobulin. It's an antibody. Plasma cells, they have an antigen binding site, the same as that came from the B cell receptor. They're specific for that antigen, and they're identical to the B cell receptor. They have a variable region and a constant region. The variable region is just where they bind. Again, each one varies and is very specific for what they can bind to. It's the hands, variable. This one looks like circles. The rest of it is called the constant region. But antibodies look pretty close to each other. They all look like a Y. The main thing that varies is just what they attach to up in that hand. And there's different ways to how they can get rid of different pathogens. So they're going to bind to the antigen at that hand, that antigen binding site. They can bind to bacteria, they can bind to toxins, and they can bind to viruses that are extracellular. What does it mean if it's extracellular? Inside or outside the cell? Outside the cell. So it can bind to viruses that are outside the cell. It cannot bind to viruses inside the cell because they're inside the cell. And it does not know how to recognize an infected cell. Wednesday's lecture with the adaptive immune system. So the humoral immune system can only recognize extracellular pathogens. Only recognizing to get rid of things that are not hiding out inside of our cells. The antibodies cannot recognize by themselves something that's inside of our cell. But my spoiler, if the humoral immunity recognizes extracellular pathogens, the cell-mediated immune system recognizes intracellular pathogens or things that are hiding out inside of our cells, which is harder to do. But those antibodies, yes, they're going to recognize those foreign things. They're going to attach to them with their hands with that little variable site. And some of the things they try to do is they try to increase phagocytosis. They want to get that foreign thing eaten. Or they can inactivate pathogens. They can inactivate toxins. But they do it in several different ways, which is my next slide. The way that antibodies can function is through something called neutralization. So again, the antibody, it's got two hands. It's got two binding sites. Those binding sites can bind to toxins or bind to specific proteins or structures, and they neutralize it, meaning that bacteria can't cause harm, that virus can't attach to cells, that toxin can't damage the cell because those foreign things that it's attaching to are now covered. So it neutralizes what it can do. So it binds to some specific part of the toxin, the virus, or the bacteria, and it allows that thing, not allows, but it prevents those different things from doing whatever they're supposed to do, whether attach or whether it's toxins that are going to cause damage. So it neutralizes how they can damage the body. Another way that antibodies can work is something called opsonization. Opsin, anything that has the word opsin in it means phagocytosis. So opsin is anything that's going to deal with phagocytosis. So opsonization is these antibodies are going to recognize these little green triangles, and the antibodies are going to coat an organism and make it, it's going to increase phagocytosis mostly by making it more readily seen and easier to eat. It usually helps when bacteria have capsules on them. If a bacteria has a capsule, we already went over to a lab and lecture earlier. Capsules try to decrease phagocytosis. They provide camouflage because the cells themselves have carbohydrates on the outside of it, on that capsule, and they're not recognized. But they can also be a little slippery, that phagocytes can't get around that bacteria that has a capsule, and it can't ingest it. But now a bacteria is completely covered in antibodies. It's not really camouflaged anymore. It's like a big red flag for our white blood cells to be like, hey, here's something cool and big that you should come over and eat, and it will. Also, it's not very slippery if it's got these spiky little antibodies sticking out all over it as well. So just by having the antibodies coat the outside of a cell triggers our phagocytes to eat it, and it's easier to eat. Another way antibodies can work is called agglutination, and it's got the word glue in it for a reason. So an antibody does have two hands, two binding sites, and it can attach to two different bacteria. Those different bacteria, they're the same kind of bacteria. They each would have, the same kind of antigen, whether it's a little red triangle or a little brown circle. And the antibody could attach to two different bacteria, two different separate bacteria. And that antibody attaches to two, and the other one antibody attaches to two, and it gets to the point where one single bacteria, which could be super, super tiny, is now attached to another bacteria, attached to another bacteria, attached to another bacteria, and you have a big clump. Agglutination literally means you're gluing together. Those antibodies are clumping a whole bunch of bacteria together. Well, that's great for our immune system. One, they're not hiding anymore, because if they were small and not easily seen, they're not hiding anymore when they're a big old clump of bacteria. And so they are more easily able to get phagocytosed. But they also can get filtered out by our spleen, which is part of our immune system. And so our spleen, that normally some of these bacteria might be able to just go right through, maybe not even get recognized, they're going to get stuck. They're going to get filtered out. And so they're no longer going to be able to circulate through our body. Another way antibodies can work is that the hand can attach to that very specific epitope, but the body of the antibody can still attach to something too. It's not just the hands that are doing everything. The body of the antibody can actually attract another kind of cell called a natural killer lymphocyte. That's where the NK comes from. Is we have lymphocytes, lots of types of lymphocytes, that do different things, and this antibody attaches to this bacteria, and the body of the antibody attaches to a natural killer cell, and based on its name, a natural killer cell kills. And so this natural killer cell can kill this bacteria. It usually kills bacteria by inserting channels or pores into the cell where they're not supposed to be pores. and it can kill the bacteria. So the antibody doesn't kill it itself, it just recruits something to kill the cell. So it's called antibody-dependent cell-mediated. It just means this antibody is going to bring another cell, and that cell is going to be toxic to the bacteria. So really long. That I don't have a picture for is that some of these antibodies can also activate what's known as complement, which is chapter 14 that we watch and listen to, and activate inflammation, also part of 14. And so these antibodies can trigger our innate immune system to be like, we could just use every help that we can get. We would love some complement proteins to help us out. We'd love some inflammation to help us out and try to get rid of the bacteria. So the antibodies can trigger our innate immune system as well. 5 antibody classes. So again, I said antibodies are also called immunoglobulins. And although antibodies are all very unique in what they can recognize, based on the body part, not the hand part of the antibody structure, we group them into one of 5 different types of antibodies. So it's based on the body of the antibody, not the hands that are specific. So the first type of antibody class is called IgG, and these are all Greek letters, so it's immunoglobulin gamma. And IgG are the most common antibody. If we did a blood draw of all of you, we would find antibodies in your blood right now, and the majority of them would be IgG. They are the longest lasting. They're found in the serum. This is the liquid portion of your blood. And it's these that can also cross the placental barrier. And so developing fetuses are getting antibodies from mom. Most common. IgM, M from you, I would not ask you to know what the different Greek letters stands for. This is the first antibody that's made. Not the most common, but it is the first one made. So if you got sick, you got some type of bacteria, virus, whatever you got sick from, the very first antibody that our plasma cells are going to spit out are going to be IgM. So they're called the first responder. But the most unique thing about IgM, not just that it's the first one that's made, is the shape. Because IgM antibodies always hang out in groups of 5. And so they look like this. I always think they look like a big old snowflake. So it has 5 bodies, and because every body of an antibody has 2 hands, 2 arms, there's 10 possible places that it could attach to. Call it a pentamer because there are 5 bodies altogether. So 5 antibodies always linked together, 10 arms. These are amazing antibodies at agglutination because you have these things that look like big old snowflakes that can attach to 10 bacteria all at once with just that one antibody grouping. IgA for alpha, these are the antibodies that are always associated with secretions. So the fact that if I collected your sweat, I'm not going to, and I looked in your sweat, you would actually have antibodies in your sweat. Any type of secretion that comes from your body is going to have antibodies in it, and it's always going to be the IgA. So it's associated with body secretions to any open surface whatsoever. Are going to have these. Your saliva is going to have antibodies in it, IgA. Your tears are going to have antibodies in it, always will be IgA. Breast milk and colostrum are going to have antibodies in it. It's one of the #1 reasons why they say breast milk is best, is because babies who do not have a well-developed immune system yet, they can't do all of this right away. But if they are drinking breast milk, they are actually getting antibodies from mom. So if mom was exposed to something, they're getting those same antibodies from mom. They're just IgA. Even semen and vaginal secretions, any secretion to the outside is going to have the IgA antibodies. IgE, these are for epsilon. These are the antibodies that are involved in allergy responses. So if you're allergic to something, if you have even seasonal allergies, you can blame these guys for your seasonal allergies. It's these antibodies that just go crazy in the spring or any other time throughout the summer or fall. And they're also involved in parasitic worm infections. Now, chapter 14, the eosinophils, which is another white blood cell, also starts with an E, are also involved in allergies and parasitic worm infections. So anything that starts with the E's, allergies, parasitic worm infections, eosinophils, antibody E. And the last one, IgD for delta. These are an antibody that actually still stays on the B cell. They don't float around all by themselves, as most antibodies do. So they're almost going to look just like a B cell receptor. The antibodies do look like B cell receptors, but it stays intact on the B cell itself. It says we don't know the exact function of these B cells. We're getting a little bit more information as we're doing lots of research, because as scientists, we don't like to not know something. And we do know they are involved still in neutralization. We know they're involved in agglutination. So we know some of the stuff they're doing. They're still doing stuff. They're still in an antibody. You wanted to write notes. But I'm going to end right there, because we got through the humoral immune system, recognizing extracellular pathogens. Got all those B cells. They could become plasma cells, but if they didn't become a plasma cell, they became a memory cell. And they turn out a bunch of antibodies. Wednesday, we're going to take a look at that cell-mediated immune system and all the T cells and how they work. So I'll put an announcement out, but otherwise, take a look at the chapter 14 video. That's due tomorrow night, midnight. But tonight, if you haven't taken that disease and infection quiz, that's due tonight. So something due tonight if you haven't done it, something due tomorrow night if you haven't done it. You probably have a good rest of your night, and email me if you have any questions.